Okay, hi everyone. We'll start the seminar now. Welcome to the seminar. This one is being hosted by two groups here at the University of Liverpool, the Earth Sciences Research Group and also the Liverpool group of glaciology researchers. The talk's going to last about 40 to 45 minutes with time for questions at the end. So if you're in the audience, just ask a question at the end. And if you're watching on YouTube or Zoom, please type a question in the chat or put your camera on at Zoom and we can ask the question on your behalf. So I'm now happy to introduce our speaker, Chris Stringer. Chris did an ME-Sci in Geology and Physical Geography here at Liverpool and focused his dissertation projects on glacial sediments and in his third year and carboniferous river sediments in his fourth year. He's now a second-year PhD student in the School of Geography at the University of Leeds, where he's researching landscape changes in Antarctica's proglacial regions. And today, Chris is going to talk to us about his PhD project and a recent exciting expedition. So over to you, Chris.
Thank you very much. Thanks, Caitlin, for the introduction. Thanks to Grace Giro for inviting me over and also to Bob Duller, who be here today. So yeah, as said, I recently got back from an expedition with the Czech Antarctic Research Programme to James Ross Island. So I'm going to talk you through some of the work I did out there and also my broader PhD project. So we've had a quick introduction to me already, so I won't go over this too much. But as Caitlin said, I studied at the University of Leeds for 4 years, researching the wide breadth of geomorphology, geology, and particularly focused my work on sedimentology. And since 2020, I've been a PhD student at the University of Leeds, working with Jonathan Karavic, Duncan Quincy, and I also have an external supervisor, which will make more sense as to why I went with the Czech Antarctic Research Programme, who's Daniel Liebel, who's head of the programme over there. And yeah. So just to quickly give you an overview of what I'm going to talk about today, I'm going to introduce you to the research I'm doing as a whole as part of my PhD, and tie that in as well with the purpose of my field work going to Antarctica. I'll talk you through the data I collected while I was out there, and also I want to end with about 10 or 15 minutes at the end of what it's actually like to go to Antarctica, what it's like to live there, work there, and really being in quite a strange environment. So I tried to sum up what my PhD is in one nice, pithy little sentence, and it turns out that that's a really quite difficult thing to do. But in a nutshell, I'm investigating land cover change in Antarctica's proglacial regions. What I mean by proglacial is pretty much anything not covered by ice in Antarctica. I did initially start to call them ice-free areas, but permafrost specialists I think a little bit upset with me for saying that. And that really, in terms of my PhD project, can be broken down into three sort of neat components. A remote sensing component, where I've been trying to characterize the land cover of the modern proglacial regions in Antarctica. And then also some work that I'm going to be moving on to in the future is to investigate decadal scale change in land cover. I did start doing some of this, but I found a better method for approaching this, so I'm putting the change detection on hold for now and coming back to that. And I also have a fieldwork and a lab work component. Now, I've separated these into two sections, but really they're very interlinked. The fieldwork was essentially collecting lots of sediment samples, river samples, rock samples from across two catchments in Antarctica. And the lab work, which ties onto that, is effectively the analysis of those samples, which includes geochemistry analysis, granulometry, and also some measurements of suspended sediment load in the rivers. And it's kind of a strange thing to focus your research on Antarctica and not to focus it directly onto the glaciers. It tends to raise a few eyebrows when I explain it to people. But the rationale for caring about this and why I would like to convince you all to care about this is that sediment plays a really, really important role in controlling the types and variety of life in marine environments, and actually indeed in lake systems and lacustrine environments. And this ranges from things like sediments mechanically damaging organisms or clogging up, like sponges and pores, but also it can just more simply be that it can block light out from photosynthesis or make it harder for animals to mate. And it's particularly important to study these in Antarctica and more generally in polar regions, because life there is particularly sensitive to change. And there's also just this fantastic variety of life in the Southern Ocean, some of which we are yet to explore. So just from the sort of explorer in me, if you like, I think it's really important to understand the controls on these systems. But if you want to go down a sort of economical route, they're really also quite important fisheries. And the reason for focusing my work not only on land cover change, but especially on river systems in these areas, is because, quite simply, rivers are the primary transporters of sediment. There is work ongoing with other people, and particularly in the Czech projects, that's looking at aeolian transport, but my research primarily focuses on the river system. And so this brings me on to the first component of my PhD and of my research. It's close to completion at the moment, but it is still ongoing, and I'm really trying to get this finished so I can get a bit deeper into the data I collected on fieldwork. But I'm aiming here to characterize present-day landscapes. I focused on 6 of the largest proglacial regions, particularly in West Antarctica. These are primarily islands. As I said, the sort of criteria for picking out these sites was primarily size, but also finding images that are color-free is a lot of work in itself, and so this also becomes quite an important factor in choosing these sites. And the idea with this is to characterize the present-day state of these regions, to really understand what they look like now, what the land cover looks like, and then therefore its implication potentially on sediment outputs. And I also want to look at the spatial variability. And the spatial variability is important because the further north you are, quite simply, the warmer it is. And if we can understand what this difference in the latitudinal gradient looks like, we can start to think about what the landscape might look like in the future at these more southern regions. So when I'm talking about remote sensing and when I'm talking about producing these land classifications, I just wanted to give you a very, very quick overview of how I've gone about this. I've searched the Landsat 8 catalogue of images of Antarctica in Google Earth Engine, and I've prepared the images for analysis. When I say image preparation, this involves a topographic correction to effectively flatten out the mountains on the images and reduce the impact of reflection from mountains and hills, and run a principal component analysis over the images. I then broadly classify these images into a land class, an ice class, and a water class, using a k-means clustering algorithm. And then I'm able to use these to split the images down to land, ice, and water, and I can subdivide these into neater, more usable land cover types. And what this produces is something looking nice and pretty like that. So as I said, I looked at 6 sites. These range from as far north as South Georgia. Okay, some people will be pedantic and say that perhaps that's a bit too far north to be Antarctica, but the British Antarctic Survey covers it, so I'm counting it. And it goes as far south as down to the Dry Valleys, and includes land in between Alexander Island, the South Shetland Islands, and James Ross Island. If it rude of me to not cover the place I visited. And as I said, this makes use of unsupervised techniques. Now, what I mean by an unsupervised technique is that I use a k-means clustering algorithm. I say I want X number of clusters in the image, gives me back these discrete sections on the image, and then I'm able to interpret what the relationship is. So, for example, it's very clear that certain clusters show snow and ice, certain clusters show land, and then using some maps of the area, you can get into a bit of a nicer interpretation of what it might be. You can split it up into then several land classes, which include different sedimentary surfaces, water, vegetation, and so on. And this is highlighting the latitudinal variation in vegetation cover, quite a nice trend in increasing vegetation cover as you move further north. and also the presence of liquid water, lakes, effectively. And I'm going to leave it there. I'm writing this up at the moment, and to be honest, I could spend the whole time talking about this if you left me to it. So hopefully it will be written up soon and submitted somewhere so you can read into it a bit more. And if you are interested, you are welcome to email me. So I'm now going to move on to the exciting bit, the bit that I think most of you are here to hear about, which was a recent expedition I had to James Ross Island with the Czech Antarctic Expedition. We were staying at the Johan Gregor Mendel station, a nice little small station on the northern tip of James Ross Island, and I'll get into it. So I want to start at the beginning, really, with the big question: how do you get to Antarctica during the pandemic? And it's with great difficulties is the true answer. And really, this is the busiest slide in the presentation. I tried to keep the amount of text in my presentations down to a minimum, but there were so many hoops to jump through to actually just step foot in Antarctica in the first place. And actually stepping foot in Chile in the first place was the biggest hurdle. It involved taking a PCR test before travel and a lot of paperwork. I was losing my mind with the paperwork because they wouldn't accept a British passport, a British COVID passport. So I had to apply for one of those a months in advance and submit everything, and it got quite difficult. But I made it. I got out of the UK and first went to Madrid, where I met up with the rest of the expedition. And then we flew on to Santiago, where we spent 20 hours and had another PCR test. Now, when I say we spent 20 hours in Santiago, that didn't mean we got to check into a very nice hotel and sleep, because if we left the hotel in Santiago, we would have had to spend 10 days quarantine in Santiago. And we didn't want to do that. So we had to stay between the international airport and the domestic airport in what was basically the entrance to the airport. It was really quite uncomfortable. But then we were able to get on to our hotel in Punta Arenas, where we quarantined for 16 days in the end. I think it was 10 days was the legal requirement for going to Antarctica. I think Chile itself had 5 days of quarantine, 10 to step foot in Antarctica. And we had to have 2 more PCR tests and a lateral flow test, and they were the ones that went right to the back of your throat. They were really quite unpleasant. But we managed to jump through all these hurdles and got onto a flight to King George Island at the second time of trying, because it turns out that there's quite a lot of wind in Antarctica. I don't know if we would have thought of this, but it makes it quite difficult to fly there. But the second time of trying, we got there and jumped on a ship to James Ross Island. So here is your first of many nice pictures. And I'm going to talk you through what I was actually doing there. And there's a theme here. I like my nice pithy sentences to sum up what I've been doing. In a nutshell, I was trying to identify and quantify the sources, sinks, pathways of sediment from 2 Antarctic river catchments on James Ross Island. And to break that down into its components, this involves using geochemistry to look at the characteristics of sediments in different parts of the rivers and indeed the rock types around there. And I complement this geochemistry technique with infrared spectrometry. Without getting into too much detail, this is in effect to look at some of the mineral properties of the sediments and rocks. And unlike other studies, this research doesn't just focus on what's coming out at the end of the rivers into the ocean. It also looks at the components that contribute to that final output. It looks at the different rock types, it looks at the different tributaries. And so this is what sort of makes it a bit different from what's been done already. And my idea with this is that if we can start to look at contributions of tributaries, the contributions of different land types, we can start to think about the sediment output of proglacial regions on a more broader Antarctic scale, rather than just being a sort of local study on 2 small rivers. And to link this back in with what I've been doing for the last year and a half, and why it relates to land cover mapping. The reason is land cover is a major control on sediment yields. The different erodibility of rock types and soils is one of the fundamental controls on how much sediment you can let out into the ocean. And as I said before, rivers are the primary transporters of this terrestrial sediment. So I've got a few different transitions going on here. Not quite standardized. So to talk you through where I've actually been, I've been at James Ross Island, which you can see for those that are actually in the lecture theatre, is on the northeast of the peninsula. And we were based on the northern tip of what's called the Ulu Peninsula, which is a large proglacial area. It's the second largest in Antarctica. And I was focusing on two river catchments there, the Algal catchment and the Bohemian catchment. And my research approach here considered two components: the contribution of different lithologies to the output of the two rivers, and the contribution of different parts of the catchment to the total sediment. To achieve this, I measured suspended sediment concentrations of discharge at 5 different sites across the two catchments to give us a sense of the temporal and spatial variability in this sediment. And I also geochemically analysed rock and sediment samples from throughout the catchment. So I am in the geology department here, so it would be wrong of me not to do a little bit of geology here. So just to give you a quick overview of what this looks like, James Ross Island is primarily the part that I was at, is primarily composed of soft cretaceous strata, mudstone and sandstone mostly. And it's really strange actually, because it feels like it's fresh mud. There are some parts that are really well liquefied, but you're walking along it, sinking in. And this is actually, really old stuff. And in part, these are overlaid by different rock types. In some places, it's overlaid by these basalts and hydroclast like Glaciers, and in other areas, it's overlaid by more recent Thermic sediments, glaciogenic, submarine sediments, and this kind of thing. Which moves me quite nicely on to my rock sampling. Now, there's a lot to take in on here. You can probably see these sort of pink pinpoints dotted all over. It's not too important to take in exactly where they were. What I want you to get from this is that I've taken samples from across both catchments from as many rock types as I can get access to. And actually, when I'm saying rock samples, I'm using this term quite liberally. You can see in some places that the samples were taken quite linearly, and these are river cuts. But in other places, there wasn't too much exposure. So actually, in this map, it also describes soil samples uptaken in superficial sediments. And the river sampling takes two parts, as I mentioned before. It takes these regular measurement sites, which you can see as red dots. And I'm not sure how well it shows up, but there are these crosses along the rivers as well. And these are bedload samples. These were just taken once. And the method for taking these samples wasn't particularly rigorous. It was sort of a grab sample from the base of the river. And what I want to look to do with these is look at their geochemistry as well, see if there's any trends downstream changes in the composition, and also look at their grain size characteristics. Sorry, I forgot to say before, these regular measurement sites, that's why I was taking the suspended samples and the discharge measurements. So in total, I came away with bedload samples, I think 40 in total, suspended sediment samples, I've not counted them, but there were a lot, and lots of rock samples as well, I think 60 rock samples in total. So I think I've got the data there to create a good characterization of what the rock types look like in the area and what the sediment looks like at different sections of the river. And this is where I'm going to say a big thanks to my colleague in the Czech Republic, Charles University, Andrei Nedelchev, for his help with collecting the data. I'll explain in a moment that it really would have been quite difficult to have done the regular measurements I took without his help. And that's because when I was taking regular samples. I was going out every 2 weeks to measure over 24 hours. Each loop of these, if all sections of the river were flowing, of course at times they were frozen, was a total of 12 kilometers round. It would take about 4 hours to walk. And as I said, I did this every 6 hours. So there wasn't much time for rest and there wasn't much time for sleep. I do this, no matter the weather. So he would, for most of it, took the 10 p.m. shift, which allowed me to get a few hours sleep, and means that I'm here and not passed out of exhaustion somewhere in a river. And all of this brings me quite neatly on to the lab work I did. I've done most of this actually at the station. I brought a handful of samples back to do the granulometry, but all of the geochemistry and infrared data I took while at the station. Now we had 2 labs. We had a wet lab. What I mean by wet lab is it had a tap, and a dry lab which didn't have a tap. And I did the sample preparation in the wet lab, and this involved sieving the sediment samples. So I extracted the sub 2 millimeter grains. The reason for doing this was sort of in 2 parts. One, I wanted to run them through a laser granulometer, limits you to 2 millimeters. It's also the most active part of the river sediments, and I also had to limit the amount of samples I could bring back in terms of weight. So choosing this fraction generally made sense. And with the fissile rock samples that I collected, so these were superficial sediments I collected, but also sort of like flaky mudstones, I crushed them up using an agate pestle and mortar, which was tough going. It was about that big, so it was hard work. The reason I chose an agate pestle and mortar is it's 7 on the Mohs scale, so it's harder than most minerals out in the rocks of the river samples. For the more resistant rock types, so your basalts, for example, I used a masonry drill. It was actually something that I discussed with John Boyle back in October. And I went up to the head of the station and went, "You got a diamond drill bit by any chance?" And if it was the other way around, I would have looked at me like I was crazy, but he went away and managed to find something that worked. Now, I don't know the exact Mohs hardness of this drill bit, but I'm certain that it's harder than the mortar. Scratched on the back of it to test it out. And most that I've read since suggest it's about 8.5. So that covers the bedload samples and the rock samples. With the suspended sediment samples, I filtered them using a vacuum filter and dried and weighed them in the traditional sort of suspended sediment method. There was also a turbidity meter there that was taking some measurements at the outflow. And all of this was much appreciated by Bendula, who's on the right of this image. You can see that we're wearing masks, and this wasn't a COVID control, we've been through our quarantine. This is because when you're crushing samples, it puts quite a lot of dust into the air. And the beauty of working in a small polar research station is that you have to share the lab with people from across many disciplines, including the microbiologists that were trying to keep a nice sterile environment. So me coming in with my all my dirt, and for all the best people in the world, it's impossible to be clean when you're crushing these samples. And eventually she set up one of her spare agar jellies to show christening mode for those that can't make that out. And apparently, just to the top, there's some nice concentric circles. Apparently, that's a really interesting mold. So, who'd have thought it? And once I prepared these samples, I was able to take them to my homemade spectrometry lab. This is super special spectrometry lab, as you might make out on the sign there. And this involved using a handheld XRF, which was lent to me by Maastricht University in the Czech Republic, and an infrared spectrometer, which was lent to me by NERC's field spectrometry facility, which is based in Edinburgh. And they were really, really good actually for getting the set up and trained on the equipment typically used to measure spectral reflectance from leaves and biological samples. So they were really good at finding the best way to actually measure sediments and rock samples. And you can also make out a box on there, over there. And that was my homemade, like also a spectral shield to block the effect of the fluorescent lights on the samples that can affect the reflectance from the spectrometer. So to sum up the data I collected, there's quite a large amount I managed to collect while I was out there. So there was the discharge and suspended sediment data that I managed to collect at these 5 locations, and they're collected every 2 weeks, of course, for the duration of the 64-day expedition. There's the geochemical data that I collected for all the river samples, for all the bedload samples, for all the rock samples, and for all the suspended sediment samples where there was a sufficient quantity for me to actually measure. I should say with the suspended sediment samples, I wasn't able to push them up because I also want to run them through the granulometer, so we'll have to be a little bit careful when we come to analyse that data. There's also other work that's been done by other people at the station. So there's been a long-term monitoring, I think 5 or 10 years of the outflow of the rivers and the amount of sediment coming out there. So there's a turbidity meter, pressure gauge, and a velocity meter, which can tell me discharge measurements and sediment load. I think it's every 10 minutes or of that order, the outflow of the 2 rivers. And there were also there's also a couple of UAVs that were taken with us, and they were able to capture images of both river catchments. And the interesting one for me is the one taken in the Peninsula catchment. There was one taken at the start of the expedition and at the end of the expedition, so I can look at changes in architecture potentially. And as I mentioned before, I'm also looking at analysing the grain sizes of these sediments, so I brought these back to Leeds to do that bit. And the bit of research that I did while I was out there that I've not really touched on were these trail cameras, which is what's being shown in this picture here, actually. The idea with this sort of experiment, and it was a bit of a stab in the dark to see if we could get anything, was to set up trail cameras looking at 3 of the days on the Peninsula to see how frequently sediment plumes were coming off rivers. It was quite windy, there was some bad weather. I'm not sure how useful this data will be. So I put their exact use to be determined. But in the very worst case scenario, I have some quite nice time on this. Not all wasted. How are we doing for time? So I want to spend the last 10 or 15 minutes of the presentation to talk about life with the Czech Antarctic Research Programme. It really was quite a bizarre situation actually to find myself with such an international team, and as the only Brit on the expedition, it was really quite unique. So there were 16 of us, most of us scientists, but also 3 technicians whose job in a nutshell was to keep us alive. In what was a fairly small building, I'd probably describe it as a very nice hostel. There were these shared rooms throughout. There was a shared kitchen, a place where we could all eat, and a technician suite in the 2 labs. And the number one question I've been asked since I got back, no one cares about the research, they just want to know what I ate. And people are quite disappointed when I've not been eating rice and beans for the past 3 months or whatever. But actually, it worked really nicely. We cooked food on a rota. So once a week, there were 2 of us assigned what was called service. So we were in charge of cooking and cleaning the station. And there was actually quite a lot of fresh food. We took the hardier vegetables, fruits, things like cabbages, squash, apples, pears, and they lasted quite well, either out in the storage unit, these like shipping containers, or in the inside cool room. And we also had quite a lot of meat kept in the freezers, and personally this was the most difficult part for me because I'm vegetarian, but it wasn't really possible to be picky. You ate what you were given, and really that's a bit of a first world problem when it comes to eating. So for the most part, we ate very, very well, very, very tasty food. Until the last couple of weeks, there was a bit of a delay in being picked up, and so we were reliant on dried things like dried beans, dried chickpeas, and also these like tinned pork and tinned beef, which was really horrible to cook. And in the end, we had 64 days at the base. It was the second longest expedition, thanks to a delay in being picked up, but it allowed me to collect a few more data points, so I'm not complaining. And really, you know, I was with a really fantastic bunch of people. It was difficult to know what to expect. I'd only met them all once at a training session back in November, but friendliest bunch of people, really funny, and you know, we socialised well. You can see in this picture we had a football match, which apparently one of the first football matches they've had there. I mean, amazes me. Don't ask the score, my team looks. But also, like, generally, you know, we've had the guitar out on several nights, and just generally, it was really, really enjoyable. But also, from a professional sense, there were a lot of opportunities for me. On top of my own research, I was able to do a little bit with other researchers, sharing the lab with Bandula. I learned a little bit about microbiology. Just in itself, making use of those whole labs was really quite unique. It really made me appreciate having a bit of ingenuity when it comes to your work, because you know we can be spoiled sometimes when we have you know, all this really nice lab equipment, or we have the ability to order in some equipment, whereas sometimes we just had to make do with what we had, like I said before, using the box that we took for the Bluetooth speaker to protect my samples from fluorescent light. And probably of interest to the Liverpool group, I did manage to get out and do some glacier work. It was actually getting towards the end of the expedition, and I've collected most of the data that I wanted to collect. And I said to them, I've not actually been on a glacier yet. I can't go to Antarctica and not step foot on a glacier. So I've included this picture here. This is on a triangular glacier, and that's me drilling a 2-meter hole into the ablation sticks. And yeah, on top of all of this, working in that international team was really, really fantastic. It's really nice to see, you know, a diverse group of people look at problems from a different perspective. And I think it's really helped me understand my own work more thoroughly. And as I mentioned before, there's also this drone data, these turbidity meters. And actually what I didn't mention was the meteorology data that I've also been granted access to. So there's this fantastic network of weather stations all over the island that I can get the data for if I need it. And I was also allowed access to the technician's woodworking station. And so the coldest place on Earth has a sign in Antarctica, Oldham Athletics Boundary Park. I couldn't go all the way there and not leave a mark. And I can't think there are now many 5th division sides that have a sign up there. It's very depressing to say 5th division. So I'm going to step back. So yeah, that was a video put together by the expedition for the press conference back in the Czech Republic when they returned. And that little bit at the end with those bizarre looking buses was when we were taken in by the Uruguayan expedition on the way home. The ship that was supposed to collect us broke down, so it could only take us as far as King George Island rather than across the Drake's Passage. So there were a few panicked emails sent out saying who can take us. And the Uruguayans very, very kindly took us in, and we were able to fly back to South America. So I like to end with that video. I think it sums up nicely what I did for 2 months. And I'm happy to take any questions you have. And thanks for coming up and listening.
Thanks a lot, Chris. That was really interesting to hear about your teaching work and the expedition. So yeah, do we have any questions either in the audience or people watching online? Yeah, I'll just bring the microphone over.
Speak back into you.
Yeah, thanks. So I'm a glaciologist. I'm an artist in glaciology rather than. So I mean, I guess right at the start, you kind of talked about the motivation. Well, part of the motivation is that so many parts of the ocean, conservation is important, and you did look at kind of the discharging of the river. I guess my question is how important the masses are to the amount of sediment flux into the ocean, you know, compared to some of the other stuff that you're looking at, in terms of rock type and yeah, those kind of factors.
Yeah. So it depends where you are. On the two rivers I was looking at, actually, they're mostly deglaciated. So the primary input of them is from permafrost melt from the active layer. In other areas, it depends where you are, but typically it's the active layer that's the most important contributor to the water in rivers. But of course, if there's a large glacier nearby and it's a warm day, it will be contributing quite a lot.
Yeah, okay, yeah, interesting. So I guess I kind of put up questions to relevant, presumably this is going to change into the future. Is that an aspect that you're looking at, or is that kind of the last part of a discussion?
Yeah, I think it's part of a wider discussion. But yeah, for sure.
Thanks for the great talk. Yeah, the field was very interesting. So I'm not a geologist or a geologist, so my question is going to be more high level. I was really interested by the subject at the beginning as well. They were talking about doing remote sensing in very large scale things, and then you had all the fieldwork. So are you planning on tying those things together?
In what sense do you mean, sorry?
So for example, using the data you gather from the field to form some aspect of your remote sensing work. For example, to extrapolate whatever you learned there spatially to the satellite images to other content.
So part of what would have been nice to do, and it didn't work out because the geotagging of my photos didn't quite work, would be to use part of it as an accuracy assessment for land cover mapping. I've had to take a different approach to assessing that now. So not directly is the answer to that really. The idea is to have a sense of what these proglacial areas look like, have a case study of what it's looking like on the ground in reality, and tie it together in that way. But they're sort of two parts that are looking at the same wider concept, if that makes sense.
Are there any more questions in here in the lecture theatre?
It looks like a huge amount of things now. But my question is about you.
Okay.
So you said that the valleys are mostly glaciers. Are they still raining?
Yes. If I have the picture here. I think certainly the picture that was on the poster advertising this, you can sort of see the extent of the grading. Don't know if I actually have it in this presentation, maybe catch it on this one. Oops. You can maybe make it out on that photo. That's the Bohemian River, and there's these huge great plains. But I can send you a photo. It's not flowing. There tends to be like that's sort of the main channel running through.
And you know how long it's been designated for?
So about 12,000 years actually. They've been deglaciated quite a long time. There's this really sort of strange thing where, because it's on the east side of the peninsula, you've got the mountains that run down Antarctic Peninsula and through Grahamsland, and they act as a blocking force for the warm, wet, fronts that come across. And so you end up with the situation that James Ross Island, compared to, say, the South Shetland Islands, are much, much colder and much, much drier. It's actually semi-arid, strictly speaking, this, because there isn't a whole lot of precipitation, and a lot of the precipitation is actually just blown off into the sea. It's snowfall.
So, is the, if the rivers are broken without direct glacial contact, what's driving the breaking? And is it dependent on glacial interaction? Is this like how are glacial adjustments, or would the rivers be breaking without that being there? No.
Yeah, good question.
It's not a question you can like answer right now, but
yeah, it's interesting.
That's kind of how it again.
It's certainly fed on a temperature scale as it would be with the glacier. I said it's based on the active layer and the amount of water that's melting off that, and actually a snowfall melting. So it's somewhat precipitation driven, but it's mostly permafrost degradation. Basically. Yeah.
What an interesting description.
Yeah.
Cool. That's not bad. I have a question about the remote sensing and the clustering.
Yeah.
What variables did you use in the clustering analysis?
I can email you that. I can't remember off the top of my head.
Was it things like colour?
Sorry. I see what you mean. Sorry. So initially you can do the first. So as I said, I've done it in two orders. I've done first ordering, second ordering. First ordering, you can do it visually. You can quite easily spot water from ice. From land. And then the second order, one of the things that's really come out of this is that there is very little ground data in Antarctica. There are very few good quality maps. So from the limited maps that are available, I was able to interpret what the clusters are. So, for example, one of the clusters I have is what I've called coarse or wet sediment, because I was seeing a relationship in the clusters between rivers and great plains and ski slopes, for example. And actually, when you go into the data and have a look at it, you can see that there's a higher NDWI index, a higher water index for these pixels, and a lower reflection, which I've interpreted as a higher surface roughness, which you can correlate with being blockier sediments, for example.
So do you verify your clustering analysis with other existing maps?
So I interpreted the clusters using existing maps, and then I validated that by going through manually to several points and looking at higher resolution images from Sentinel to see if it's correct or not.
Okay. Are there any more questions? Do we have any more on Zoom? Or on YouTube? Okay, well, if there are no more questions, I think we'll end it there. So thanks again to Chris for a brilliant seminar today and for coming all the way from Leeds to give it. And thanks everyone for coming online and in person. So now we're going to the next one.
Excellent.